My name is Chirath Easwarage, and today we are going to cover immunity. So I'm going to strictly stick to the resource book, and at the same time I'm going to show you some diagrams so you can have a visual idea, right? Because at the end of the day, what you need is the resource book knowledge in order to get a good A pass, and also sticking to the resource is going to save you a lot of time, because immunity is actually it's a very small content area, which is something that students don't think of. And at the end, we are going to do some past paper questions, right? And yeah, so that's what you can expect in this video. So let's get into it. So I'll start with the resource with some definitions that we have in there, right? So the first definition that we have, right, I'm going to highlight definitions in this color, right? So the overall general definition of immunity that you have to know is the state of being resistant to injury, invading pathogens and foreign substances through defensive mechanisms in the body, that is, you are immune, right? So then you have a definition of what are these pathogens, right? Pathogens are the agents that can cause infectious diseases, and you have to know what are the categories of pathogens, right? So they can be bacteria, virus, and fungi, right? Sometimes it's not said here, but protozoans also can rarely cause diseases, right? Then, so anything that is recognized by the body cells as non-self, that does not belong to your body, can be called as a foreign substance, right? So other than your pathogens, right, some substances such as pollen grains, incompatible blood cells, and transplanted tissues can also be recognized as foreign substances, and therefore they will also induce a defensive mechanism in the body, right? An immune response in the body, right? So we have to know that, so the immune system must be have the primary ability of identifying who are my cells and who are these foreign cells, foreign substances, or foreign cells, which could be a pathogen as well, right? So they have the ability to distinguish own body cells that are self cells from foreign cells or particles that are called as non-self, right? Okay, so there are special cell types in immunity. We'll get to that, the classifications, right? And also there are several chemicals. Okay, right. So let's get into the classification, right? So immune response we can divide basically as innate immunity and acquired immunity, right? Innate immunity, let's get into it. I'll explain with diagrams as we go, right? So innate immunity definition is the ability to resist damage or diseases in the body through inherent body defenses, which offer rapid responses against a broad range of pathogens and foreign substances, right? So what I need you to put into your minds is this term broad range of pathogens, right? So because of this broad range, this innate immunity is sort of a jack of all trades. It's going to give you a general protection against anything that is foreign to your body, right? So that's why we call this innate immunity as non-specific immunity as well, right? So the other part, or the opposite of this immunity, is the other type of immunity that is mentioned here, or acquired immunity, or adaptive immunity, that we can call also as specific immunity, right? Okay, let's continue under your innate immunity. So the recognition and defense responses of your innate immunity will depend on characters common to groups of pathogens, right? So it depends on common characters, right? Then innate immunity lacks specific responses to specific invaders, and its protective mechanism function the same way regardless of the type of invade. So imagine your innate immunity as sort of the ready army in a country, right? So that army is always available to respond to any type of a threat, right? But that army is the only army that is ready at the moment to respond to any threat. It is not specialized to target any specific enemy. It's going to provide you a defense against all enemies, right? So it's not an expert against one type of enemy, but it can give you a general protection from all enemies, right? It's like the always on duty army in your body. Okay, think of it that way, right? So then what you have to know that innate immunity is found in both invertebrates and vertebrates, right? Okay, now let's go into the categories of innate immunity, right? We can categorize innate immunity as your external barriers and internal barriers, right? Okay, so as I mentioned, so this innate immunity is always there in your body to provide defense. So it is going to have two lines of defense. So the first line is the one that is going to protect you against the first wave of enemies that are coming, right? So it's going to be the outermost covering, so you can imagine. Then if the enemy goes through that, then there's going to be a second line of defense of innate immunity that is going to give you some protection. Okay. Right. So innate recognition. So I mentioned that innate immunity is going to recognize pathogens based on common characteristics, right? So such as this is kind of that. So if it's a virus, it's going to have some recognized molecule, right? And it's a bacterium, such as, then it could be a protein such as flagellin. We are going to come into this antigen term later as we go on in the video, right? In fungus also, it's going to have some common character that is going to identify, this is a foreign substance or a pathogen that I need to protect. But it does not know that this is this fungus, that is this virus, or it cannot distinguish different pathogens from one another. It's going to know that this is a pathogen, so I need to give protection to the body from it. Okay, right. So your innate immunity is sort of primitive or primary source of defense that both invertebrates and vertebrates have it, right? So if we can imagine this external component that we called as the first line of defense or external defense, if you can imagine our body as sort of a city that is protected by a wall, right? Balkotu, right? And that wall itself is what we call as external defense, right? So it is the first line. So if there's an enemy invading your body, that is going to encounter that wall first, right? So keep that in mind as I explain what is external defenses and what defenses are there under this external defense, right? So let's see. So what are the like defenses in your external barrier? We have the skin, mucous membranes, and secretions, right? So we say that they act as either physical or chemical barriers. So they can be physical or chemical, or they can be both actually, right? Physical barriers are we can also call them as anatomical barriers, or like structures in our body that's going to use some physical defense, right? Just because they are there, they are going to provide some protection. But chemical barriers we also call as physiological. They're going to do something. They are going to maybe change the chemical, maybe they're going to influence or change the pathogen in a chemical way, right? So first example is your human skin. Let's read the description first. So the human skin has many layers of closely packed keratinized cell layers. So if you can remember, on epithelia we studied that the skin consists of the skin epidermis, specifically stratified squamous cells, right? And in the epidermis, it is also keratinized, right? So keratinization is going to provide protection from like desiccation, as well as it's going to be a very tough layer, right? So it's going to be the main significant, like main physical barrier, right? And also skin undergoes shedding, right? So the outermost epidermal layer is going to get removed periodically. So if there is a pathogen or some microbe population on that skin surface, that is going to get removed as well, right? So the skin, imagine that is the physical wall that is very tall and very hard to get over, right? Then then the mucous membranes, right? Mucous membranes we consider as a physical as well as a chemical barrier as well, right? So where are the locations that we can find a mucous membrane? In the respiratory tract, the digestive tract, urinary tract, and the reproductive tract. So mucous membranes obviously produce mucus, right? Mucus is very sticky, no? We know under saliva we studied the components of mucus. It contains some chemicals such as mucins, right? Mucins is sticky and traps microbes and other particles. And almost always where there's a mucous membrane, the underlying epithelium is going to be ciliated. It's going to have cilia. And what does cilia do? It's going to move rhythmically and it's going to move the mucus layer, right? Move the mucus layer forwards, right? So if we take the respiratory tract for an example, right, we call that process of cilia moving the mucus as the mucus escalator, right? So that cilia is going to move the mucus with the entrapped foreign particles upwards, right? And also in the respiratory tract, we have things like coughing and sneezing that is going to accelerate. So when you cough or sneeze, that mucus is going to rapidly come up, right? Then secretions we consider as again physical or they can be chemical barriers, right? What are the examples? They can be tears, saliva, or mucus, right? Again, mucus is considered here. Right? So tears are found in your eyes, right? We know once there is some sort of an irritant that is like hurting our eye, the tear production is going to rapidly increase, right? So it's going to do two things, right? It's going to provide a washing action that is again going to dilute your microbes. It is going to reduce the concentration. So reduced concentration of microbes has not enough power to do anything, right? So it's going to prevent settling of them because there's a protective layer or the cornea of your eye, the outermost layer. It's going to remove the microbes. Then if you take saliva, we know the protective functions of saliva, right? So it is going to reduce the colonization of microbes, right? And then mucus, we already know, it's going to provide again a continuous washing action and inhibit colonization. And then there's a significant physiological defense here by the enzyme lysozyme, right? So lysozyme has the ability to destroy cell walls of certain bacteria, right? So this is going to make it easier for these bacteria to get destroyed because the cell wall has been somewhat destroyed, right? Then in your stomach, we have a very concentrated, very highly acidic environment by your gastric juice, right? So this very high acidity can destroy many bacteria as well as other pathogens, including bacterial toxins, right? Then also on the skin surface that we have a very low pH, a sort of acidic pH created by the sweat and sebaceous glands that is also going to prevent the growth of bacteria. So imagine I mentioned that your external defenses can be like that wall, right? So the skin is the wall itself, right? If you can imagine these secretions and the mucous membrane as a water canal, diagonal. And that water canal can contain maybe crocodiles that are like similar to these lysozymes. So if an enemy is going to invade your city, they have to get through this first line of defense first. Right. So this invading pathogen or the foreign body has gotten away from the external wall, right? The first line of defense, that is an external barrier of the innate immunity, right? But now it's going to encounter another line of defense. Think of it as another layer of soldiers or another group of soldiers that are waiting just inside the wall to guard your city, right? To guard your city, right? So it is the second line of defense according to your resource, right? So now it's going to include another, the thing I mentioned earlier. It's going to now properly identify if this is my body cell or this is like an invading foreign body cell or a foreign substance, right? So it is going to involve molecular recognition, right? Molecular recognition with receptor molecules on specific cells of the immune system. I'll show you this sort of receptors on immune cells, right? So it's going to include, so I mentioned a certain group of soldiers, such as, so these soldiers are your phagocytic cells, your NK cells or natural killer cells, and it's going to include some weapon systems such as antimicrobial proteins, right? And an inflammatory response. Inflammatory response is sort of an entire process. It's an entire phenomenon that is going to focus the attack. It's going to it's going to provide to you like a battlefield where a lot of reactions, where a lot of defensive maneuvers against that pathogen is taking place, right? So we'll come to inflammatory response. We'll cover inflammatory response in detail, right? So phagocytic cells. So what are phagocytic cells? These are specialized cells that can ingest microbes, foreign particles, and cell debris, right, for intercellular digestion and destruction, right? So phagocytosis. Before coming to phagocytosis, I want to mention, so we are still covering innate immunity, right? So this part, right? So I want to bring your attention here, right? So this is the time after infection. So we are still in like the first stages of an infection. Infection is a sort of like an invasion by a foreign substance, right? A pathogen. So in the very first stages, only your innate immunity is active, right? So innate immunity, we know the external defense or the external barriers is included by this epithelial barriers, right? So the pathogen has gone through here. So epithelial barriers are out. Now it's going to face these phagocytes, natural killer cells, and complement proteins. Others are not necessary for you, right? So what is phagocytosis, right? It's this, all right? So if you can imagine that, if you can see that this is the pathogen, like the foreign substance, right? So the cell is going to eat it, right? By endocytosis, cell eating, right? So pseudopodia are like in an amoeba, you know the process that they intake food particles is by this pseudopodia forming. Similar in your phagocytic cells, right? It's going to form a food vacuole, right? And you know, under your cellular basis, you learned about lysosomes. Lysosome contain digestive hydrolytic enzymes. So it's going to fuse with this food vacuole and it's going to release its enzymes. So the enzymes are going to destroy this pathogen, right? And it is converted to debris. And those are given out as exocytosis, right? Out of the cell. So this is phagocytosis. We have two types of cells in our innate internal immunity that is going to do phagocytosis. Those are neutrophils and macrophages, right? So out of these two, neutrophils are the people or the cells that are first attracted to the infected site, right? By signals from infected tissues. These are chemical signals. Therefore, being attracted somewhere because of a chemical signal, we call it as chemotaxis, right? Chemotaxis. So this chemotaxis is because of a type of chemical that we are going to learn in future under antimicrobial proteins. We call it cytokines. Cytokines, right? So cytokines are of many varieties. We call them interleukins or interferons, right? You can see out of these certain cytokines, you can see here, interleukin 8, for example, involves in chemotaxis of neutrophils. So this is just an example that you don't need to remember, but I'm showing it to you to show that these chemicals, these are the chemicals that are going to attract your neutrophils to the site of infection, right? So we know, so neutrophils are the first people there. Then they are going to start phagocytosis, right? Then after that, the macrophages are going to come, and they are actually larger and bigger, more potent. So they are going to do phagocytosis in a much larger scale, right? Then we come to the other type of cell that we have in our internal defense. Those are natural killer cells. So as the name suggests, they are able to kill foreign cells and pathogenic cells, right? So they are actually a type of T lymphocyte. We'll come to these lymphocytes when you are doing specific immunity, right? They are actually type of T lymphocytes, right? They can detect cells with abnormal surface molecules, right? Depending on surface receptors, and they can kill them, right? So they are not killing these cells by phagocytosis, but they are using certain chemicals to kill the virus infected cells, right? So that is natural killer cells. Then we come to antimicrobial proteins. right? That is actually an umbrella term that we give in your, that they have given in your resource. So they are including interferons and complement proteins under it, right? So actually interferons are actually a category of chemical signals that are actually known as cytokines, right? Cytokines are of two types, as interleukins and interferons, right? But you only need to know interferons, right? So let's talk about interferons first, right? Interferons, they say, are proteins secreted by virus body cells that protect uninfected host cells from viral infections by interfering with the viral replication, right? So actually, it's only a few interferons that do that, right? For example, you can see interferon alpha is a one type of interferon, right? Interferon beta, right? Also, they are secreted by viral infected cells. So they are going to provide, so they are released by an already virus infected cell, and they when they go into a healthy cell that has not been yet affected by the virus, they are going to give some protection to that for now healthy cell, right? So induce to give a resistance to cells against viral infection, right? So let's see the results. So they are released by virus infected cells, and they diffuse to unaffected neighboring cells. They are stimulated to produce antiviral proteins. Okay, a key term you have to remember. They become antiviral proteins. So they are going to give these healthy cells some protection from viral infection by inhibiting viral replication or virus dividing. Right. Then also some interferons activate macrophages which enhance the phagocytic activity. Right. So it is also given here. See, interferon gamma is involved in activation of macrophages. So you have to remember that as well. Right. So remember, they have given these under interferons, under antimicrobial proteins, but actually they are a type of chemical signals we call as cytokines. Right. Then we come to the next part under next type of chemicals under antimicrobial proteins. Those are complement proteins. Complement proteins. So what are complement proteins? They say they are a group of normally inactive proteins in blood plasma and the plasma membranes. When they are activated by different substances present on the surface of microbes, a cascade of biochemical reactions occurs which leads to lysis of invaded cells. Right. So where else do you know of a cascade of reactions? In blood clotting, right? So it is very similar to that in here as well. There's a series of enzymes that we called as the complement system, right? So if the complement system goes to completion in response to a certain infection, it's going to induce a phospholipase, right? It's like this. So phospholipase is an enzyme that can damage the plasma membrane, right? The phospholipids of the plasma membrane. So this is a diagram indicating that. So this is a complement protein in your complement system, right? So when it is, so this is the foreign cell, right? So this is a foreign cell. It has indicators. We'll learn what are these. These are called antigens. So it's going to show that these antigens are showing that I'm a foreign cell, right? So the complement proteins are going to get attached to those antigens, and it's going to produce a pore. A pore is a path through which this pore is going to allow a flow of water and other ions, right? So water and ions are going to rush in. So that is going to swell the cell, and at the end it's going to lyse or like get destroyed, right? So this is the function of your complement proteins. In addition, they mention complement proteins promote phagocytosis and inflammatory response, which we are going to study next, right? You know what is phagocytosis already. Okay. Then inflammatory response is a story like phenomenon that we have to know. This can be an essay question as well. So you have to know in detail and in a very specific manner, right? So the definition is innate immune response in the body to tissue damage triggered by microbial infections or injury to tissues. Okay. So, you have to know what are the things that are going to happen under inflammatory response, right? So I'm going to number them. So first one, release of substances that promote increased permeability and dilation of blood vessels. 1. 2. Enhance migration of phagocytes. Number 2. Destruction of involving pathogens. Number 3. And aid in tissue repair. So 4 things are going to happen under your inflammatory response. So think of this as a battlefield, right? So the body is trying to isolate the invading enemy into a small area without allowing it to spread, and it's trying to solve the problem in that area. So it's going to put all the defenses, everything it has into a small area, right? So that area is going to be a massive battlefield. So the first thing, release of substances that promote increased permeability and dilation of blood vessels. Because most of the immune cells and molecules are in the blood system, right? So these molecules, or let's say the soldiers, have to move into the battlefield. So the doors have to be open. So the blood vessel dilation, by blood vessel permeability has to be increased, like this. So this is the diagram I'm going to use to explain inflammatory response. So imagine this splinter is the injury, and it contains this sort of pathogen cells around it, right? A usual injury, a normal injury that can happen, right? So when first then we have removed the splinter, but the pathogen cells are in our skin, right, in our body. So here, this is the normal situation. This is what's going to happen. This is step 1. Can you see? These immune cells are now going through the pores or the pores that are open in blood vessels. So that is because of increased permeability. That permeability was increased due to these signaling molecules mentioned here, right? Then second thing, enhanced migration of phagocytes. That is what's happening here. So you can see it's labeled. These are neutrophils, the purple color ones. So that is happening. Migration is happening. Right? So the first thing that is going to happen is phagocytosis. Right? Then third thing, there's going to be destruction of invading pathogens. And final thing, tissue repair. Right? So this is what I mentioned. The inflammation is trying to localize the problem and prevent it from spreading to other tissues. So it's going to keep the problem at one place without letting it go everywhere, right? Okay, so inflammatory response starts with various signaling molecules upon infection. So you have to know some of these signaling molecules. That is, one of the important ones is histamine. Histamine is secreted by actually your mast cells. Mast cells, you have learned that in your connective tissue or the loose connective tissue, we can find mast cells, right? Mast cells secrete histamine. So what does histamine do? Histamine is going to dilate, increase permeability and dilate nearby blood vessels. So usually near a tissue, it's going to be blood capillaries, right? So increased permeability is going to do what? It's going to enhance the infiltration of white blood cells, antimicrobial proteins and protein elements, all the important things we require for this fight, right? And what's going to do? What's the purpose of dilation? So dilation is going to increase the diameter of the blood vessel so that many defensive molecules or cells can come into the site. So more blood can flow through the damaged area. Right. So first, the phagocytes are going to filter from the blood into the damaged tissue area. Right. So phagocytes can also discharge signaling molecules. So these are the cytokines I mentioned earlier. So that they include interferons as well. Right. So they are going to promote blood flow to the injured and infected site. Right. Then again, complement proteins we mentioned here. Right. Complement proteins can cause inflammatory response as well. So that is mentioned here again. Complement proteins can cause further histamine release. So histamine is going to again increase permeability and increase cause dilatation. So these are done by systemic, right? So more and more phagocytic cells, phagocytes will enter the tissue site, so the damaged site, and further phagocytosis will be carried out, right? Then how do we know that in a certain place of the skin there is an inflammation taking? It's edema, right? In semen, right? So what are the typical features of edema, right? Or a site of inflammation? Redness, heat, swelling, and pain. Then you have to know what are the reasons for each of these. So, what is producing the heat, right? Heat is produced due to the high metabolism. Heat by high metabolism, right? You have to know the feature or the sign and then the reason. Then what is causing the swelling? Swelling is caused by increased permeability of blood vessels, right? Increased permeability means, right, there is a lot of fluid leaking from the blood vessels or the blood capillaries. So there's going to be fluid accumulation in that inflammatory site, right? Then pain. Pain results from injury to neurons by microbial toxins, right? It's by microbial toxins. So toxins are going to damage cells, right? That includes neurons. So when neurons are damaged, they are going to generate a stimulus that we can sense as a pain, right? So as a result of, so we now know the reasons. So heat is due to the high metabolism, swelling is due to the leaking of tissue fluid, pain is from damage to injury to neurons, thereby, so that is perceived by our brain as pain, and redness is also because of the increased permeability, there's leakage of blood, right? Then pus accumulation is another feature. What is pus, right? Pus is a fluid rich in dead phagocytes, dead pathogens, and cell debris from damaged tissue. So pus, imagine like after a battle, there's going to be dead people, dead soldiers in the battlefield, right? So that is what we see in our body as pus, right? So it's going to include dead pathogens. So dead pathogens are the dead enemies. So dead phagocytes are our soldiers, right? Cell debris is the damage caused by the pathogen into our own body cells, right? So everything is going to form pus, right? Right. And then so depending on the degree of this enemy that is invading, this inflammatory response can be a big one or a small one, right? So if it's a minor injury or a minor inflammation, it's going to cause a localized inflammatory response, right? So it is only like limited to a small area. But if the injury or if the invading pathogen is a very powerful one, it's going to cause a systemic response. So systemic response can have the ability to cause a fever, right? A fever, right? In a fever condition, what is the more basic symptom? There's elevated body temperature, right? So in enzymes, we learned that when the temperature increases, the rate of enzymatic reaction can increase up to a certain optimum temperature, right? So because of this 2-3 degree elevation of temperature, certain chemical reactions, including enzymatic reactions, may be accelerated, enhancing the defensive reactions such as phagocytosis and tissue repair. So that is the purpose of elevating your body temperature in a fever condition. Okay, so that concludes your inflammatory response. Okay, now we have arrived at acquired immunity or specific immunity topic, right? So let's just go through the resource first and get through the definitions. So the acquired immunity is the ability of the body to defend itself against invading foreign agents, which could be pathogens, through specific defense responses mediated by T lymphocytes. Specific word is very important, T lymphocytes and B lymphocytes. Then three features of acquired immunity that are very, very important to remember. Specificity for particular foreign molecules, then recognition of own molecules, or those are known as self molecules, from non-self, and the third thing, memory, immunological memory for previously encountered pathogens. such that a subsequent encounter causes a stronger and more rapid response, immunological memory. So we know innate immunity is found in both vertebrates and invertebrates, but acquired or specific immunity is found only in your vertebrates. So let's go through the resource before going to some diagrams. So the cells that are involved in your acquired or specific immunity are your type of lymphocyte, the T and B lymphocytes, right? So they arise from stem cells in the bone marrow, right? Then the lymphocytes that migrate to the thymus organ in the neck for the maturation are called your T lymphocytes, right? And the lymphocytes that remain in the bone marrow, so T for thymus, so T for T lymphocytes. Then the lymphocytes that remain in the bone marrow for completion of development are B lymphocytes. So if they remain in the bone marrow, then B lymphocytes, right? Before leaving these lymphocytes for secondary lymphatic organs could be lymph nodes and lymphatic tissues, okay? We'll come to that. Their plasma membranes acquire diverse protein receptors, antigen receptors, which have the ability to recognize specific ferrine invasions. There can be over 100,000 antigen receptors on the surface. Right. So before coming to these antigen receptors, let's know what is an antigen itself first. Right. An antigen, there's a definition, is a substance that has the ability to stimulate an immune response through T lymphocytes and B lymphocytes and react with the specific cells or antibodies that resulted from the stimulated immune response. Right. So let's come to what is an antigen first. So let's take this. So this is how I'm going to explain this in full detail later. So this is your pathogen, right? So it's small part of a pathogen, right? And the pathogen's outer membrane will contain certain proteinous components that are actually unique. It's like the identity card of that pathogen. And that pathogen, so that those particles are known as antigens, right? So according to definition, right, it's a substance. So they can be, what are the things that can be qualified as antigens? Viral proteins, bacterial toxins, and chemical components of bacteria such as flagella and cell wall can be antigen, right? And so these are parts of pathogens, right? Viral proteins, bacterial toxins are all pathogens, right? But I mentioned foreign substances can be antigenic. So structural components of incompatible blood cells, right? Incompatible blood cells, we know under circulation we talked about what are the antigens present on red blood cells, right? So if an incompatible blood type is given to a person, it can induce an immune response. Agglutination, we talked about that under circulation. And also transplanted tissues. So you can transplant a kidney, a liver, a heart, many organs, and because it is taken from another person, those tissues contain a lot of these sort of antigens that are not recognized by the recipient's body. So they can be antigenic, right? Transplanted tissues. Okay, so antigens are usually foreign molecules such as proteins and polysaccharides, right? So in general, not the entire antigen, but certain parts of a certain antigen act as triggers. So this small accessible part is what the antigen receptor actually binds to. It does not bind to the entire antigen, right? So the small accessible, or the part that can connect with the receptor is known as the epitope, right? So the definition of an epitope is small accessible portion of the antigen that binds to a specific antigen receptor of T or B lymphocyte is called an epitope. So the example they have given is a group of amino acids in a large protein can serve as an epitope. Usually a certain antigen has several epitopes, right? So it's that same diagram. So if this is the pathogen, this is the antigen, so this is an antigen, and in this diagram it's a simplified diagram. It has two epitopes. This antigen has two epitopes, right? Right. So okay. let's go into the structure of your acquired immunity first, right? Adaptive or specific immunity first. So these are the main components of that is involved in this acquired immunity. There has to be an antigen presenting cell we learn about, such as macrophages and dendritic cells can be antigen presenting cells, and there are T cells and B cells, and there are 3 T cell types that we have to know. That is the cytotoxic T cell, the helper T cell, and the memory T cells. Okay. Then there are three types of B cells as well. Actually 2 types. Okay. So the B cells will have an effective form called as plasma cells and a memory form called the memory B cells. Right. So those are the main components of your acquired immunity. Right. Now let's move on to the 2 functional parts, or how this acquired immunity works, how the B lymphocyte mediated immunity works and the T lymphocyte mediated immunity works. Okay, so let's refer to the resource again. Right, so we have 2 names for the T lymphocyte mediated immunity and B lymphocyte mediated immunity. So what the T lymphocytes does is known as cell mediated immune response. Okay, so this is the definition. It's a type of acquired immunity which specifically sensitizes T lymphocytes attached to the antigen undergo proliferation. Proliferation is rapid cell division and eventually differentiate into cytotoxic T cells. Cytotoxic T cells are actually the effector form or the working type of a T cell. So they can directly kill the cells with the invading antigen. In addition, so we know there are 3 types of T cells, the cytotoxic, the memory, and there's another type called helper T cells as well, right? So the memory T cells can cause stronger and more rapid response at the subsequent income. So the memory cells are going to remember a certain pathogen with a certain antigen. So the memory cells are, after this episode of attack or infection, the memory cells are going to stay in circulation, and again if that same antigen and the same pathogen is seen in blood after a while, the memory cells are going to recognize it, and it's going to rapidly cause this cell mediated immune response. So it's going to be stronger and more rapid, right? Okay, so cell mediated immune response is particularly effective against infective cells, right? So if a fungi, parasite or a virus is already inside body cells, T lymphocytes are the best ones to respond to that, right? The other examples are cancer cells and foreign transplanted cells. So T lymphocyte mediated response or the cell mediated immune response always involves cells attacking cells. So this diagram basically shows how this cell mediated immune response by the effector form of T cell is known as what? The cytotoxic T cells takes place, right? So an activated cytotoxic T cells that will release molecules that make pores on an infected cell's membrane, and it will also release enzymes that break down proteins, promoting the cell's death. So this is the cytotoxic T cell, and this is the infected cell, right? So you don't need these accessory proteins or anyway. So this antigen is identified by the antigen receptor. Can you see? This is the antigen, and the antigen receptor is here attached to the cytotoxic T cell. And so these enzymes, or these are granzymes, granular form of enzymes, right? Perforin is the like the pores formed on the surface of the infected cell, so that is going to cause cell death, right? So that is going to cause actually lyse. Then, so this is the dying infected cell, right? So the cytotoxic T cell will get released because its job is now done. Okay, so that is how your cell mediated response by the T lymphocytes works, right? We'll move on to the other form of immunity, What is the other type of cell in your specific immunity? We have B lymphocytes. B lymphocytes is going to do what is known as humoral immune response. Right? Humoral immune response. So the definition is it's an acquired immunity which specifically sensitizes B lymphocytes attached to an antigen undergo proliferation and eventually differentiate into plasma cells that secrete circulating antibodies that neutralize and inactivate specific toxins and pathogens in blood and lymph. Very important because blood and lymph are the fluid form of our circulating fluids, right? Like where the fluid is contained. So we say that the humoral response mainly works in antigens present in body fluids and also outside cells. So extracellular pathogens, mainly bacteria, that multiply in the body fluids. So the weapon that these humoral response use is called antibodies, right? So this, how are these antibodies produced? The antibodies are produced by the active form or the effector form of your B cells that we called as plasma cells. So it's like this. So once the immune system knows that there's a invasion or there's an antigen, it's going to be detected by the B cell, and B cell is going to differentiate into its effector form, which is known as a plasma cell. Right? Plasma cells is going to produce a weapon called as antibodies. Right? Antibodies are mostly circulated in your body fluids such as blood and lymph. Right? So the antibody mediated immune response by the B lymphocytes is most effective against pathogens in your body fluids. So how does this antibody? We'll first of all see what an antibody is. Right? An antibody is a protein secreted by plasma cells. Plasma cell is the differentiated or the effector form of B lymphocytes in response to specific antigens. The antibody binds with the antigen to neutralize, inhibit, or destroy it. Right. So antibody, a typical antibody looks like this. Right. So this part is what we call as an antibody. Okay. I'll tell you later that actually, so this is actually the B cell antigen receptor. So antigens are binding to these sites, but actually an antibody is the secreted form of B cell antigen receptor. So this part will get detached and get secreted by the plasma cells in the form of antibodies. Antibodies, there are 5 types as IgA, IgG, IgA, IgD, IgM, etc. Right? And IgE, right? So there are 5 types of antibodies. You don't need to know the types and their specific functions, right? So this is the typical structure of an antibody, right? How does an antibody work, right? So this diagram shows you that. So antibodies are circulated in your body fluids such as blood and lymph. They will bind to the antigens. So this is a virus we are taking as the example. So there are antigens on top of on the surface. Right? Either it can block the virus's ability to bind to a host cell, or it can neutralize. Right? It can make the the pathogen or this virus actually tasty to a macrophage. It is like, so it has been marked for death. This cell is good for phagocytosis. So an antigen bound to a pathogen cell, in this case a bacterium, is an indication. It's like an invitation for a macrophage or a neutrophil to come and phagocytosis it, eat it, right? So that is the other function of an antibody. Okay, so it's mentioned here. It does not directly kill the pathogens, but it can interfere with the activity of the pathogen or mark the pathogen for inactivation or destruction, right? So that's what I mentioned. It can make the pathogen look tasty to other phagocytic cells, right? In addition, antibody antigen complexes can activate the complement system. Remember, complement system if it's get completed that cascade, it can lyse the pathogenic cells, invading cells. Also phagocytosis. The other name we have for antibodies is immunoglobulins, right? It's a type of protective protein. We learn this in cellular basis as well, right? So this is what I mentioned. They have the same Y-shaped structure as B lymphocyte antigen receptors, but are rather secreted. Okay. So now we come into the story form of how from the beginning, from the first moment a pathogen enters the body until it is entirely eliminated or entirely defended, the body is completely defended, we are going to learn as a story, right? So up to here we have that, and could be an essay, the role of T lymphocytes and B lymphocytes in acquired immunity. Okay, let's first go through the resource. So, for an acquired immune response to occur, some T lymphocytes or B lymphocytes must first recognize a certain antigen is present, right? Even though there is a vast variety of antigen receptors present on different B lymphocytes and T lymphocytes, only a small fraction is specific for a particular epitope. So, what is an epitope, if you can remember? It is the accessible part of the antigen, right? Right. So, because there's a large number of receptors, it has a very low chance that a T lymphocyte or a B lymphocyte will exactly connect to a epitope. So what has to be done is for a T lymphocyte or B lymphocyte to recognize there is an antigen here, that antigen has to be presented to a T lymphocyte or a B lymphocyte. So it is what they say. It has to be presented until a match is made. Okay. So proper recognition of the antigen occurs if there is a good match between the epitope and the antigen receptor, right? Okay. Now we have to know, but this recognition by a T lymphocyte or B lymphocyte differs between B and T, right? First, let's see how T lymphocytes recognize antigens or epitopes, right? So it says only T lymphocytes only recognize fragments of antigenic proteins that are presented to them by special cells called antigen presenting cells. Antigen presenting cells, very important to remember, can be macrophages, dendritic cells, or B cells. So this is known as antigen presentation. So it is shown here. Antigen recognition by T cells. Can you see? So this is a T cell. So around the T cell there are many antigen receptors. This is the pathogen. So pathogen, these are the antigens, and these small parts, different shapes, are the epitopes. This person is your antigen presenting cell, right? Antigen presenting cell is going to take part of the antigen, and it is going to make it available for this T cell antigen receptor. You don't need to know this MHC molecules. There's another process involved in that, but you have to know there's going to be a cell called an antigen presenting cell that should show that there's an antigen to a T cell, right? So this cell can be what? A dendritic cell or a macrophage, right? Or B cell, it could be also B cell. But the B cells can recognize and bind to antigens present in blood plasma directly, right? So this is that. See, the B cell does not require an antigen presenting cell for it to recognize. See, it is going to directly, this B cell is going to directly connect with the epitope of the antigen of the pathogen, right? Clear? So that part is now done. So what we are finished, we are done with the step. Recognition step is now complete. Right, once recognition is done, there is going to be a process happening, starting with a proliferation. There is a rapid cell division, a multiple cell divisions resulting in a clone, a population of cells that are identical to the original lymphocyte. So since this clone contains a cluster of cells that originated from a single cell, so the cell that recognizes there's an antigen here, right? So there's going to be different fates of different fates to the cells of this clone. Some of the cells of the clone are going to become effector cells, right? What is the effector cell of your T lymphocyte? They are cytotoxic T cells, no? Or it can be plasma cells. There are two effector cell types. Or they can become memory cells. We'll come to that later. Right? So this is the diagram. I will come to that diagram again. Right? So this is the proliferation or the clone. See, it's a clone of plasma cells if it's a B cell. It's a clone of cytotoxic T cells if it arose from a T cell. Right? We'll come to the memory function, how the memory cells at work later. Right? So proliferation and differentiation is done. Now they are going to do their work. We now have the army. So the army is going to do their work. Right? So we know the effector forms. So the T lymphocytes have cytotoxic T cells and helper T cells. Right? We know how the cytotoxic T cells work, right? Then the enzymes and killing of cells I told you earlier. And signals from helper T cells activate cytotoxic T cells to kill the infected cells. Also, signals from helper T cells can also activate B lymphocytes to initiate antibody production. I'll show this with the diagram. Effector forms of B lymphocytes are the plasma cells, right? So, a single activated lymphocyte can produce thousands of identical plasma cells. So, the plasma cells begin producing and secreting a soluble form of the lymphocyte antigen receptor we call as an antibody in large quantities. So, the circulating antibodies can neutralize and inactivate the specific toxins and pathogens in body fluids. So, this is the elimination step. Okay, the last step is providing immunological memory. So, I mentioned the clone of cells arising from the single sensitized T cell or the B cell, right? So, if it's a T cell, we are getting memory T cells, and from the B cell, we are getting memory B cells, right? Both serve the same function, that is to provide immunological memory. So if the same antigen of the pathogen or the foreign substance is encountered later in life, these memory cells are going to remember, we have encountered this pathogen earlier,